Chapter 21, The Digestive System. This chapter explores the structure and function of the digestive system, a vital network responsible for breaking down food, absorbing nutrients, and eliminating waste. We begin with the anatomy of the digestive system, identifying key organs and their roles in digestion. Then, we examine the functions and processes that facilitate food breakdown, secretion, absorption, and motility. The regulation of gastrointestinal function highlights how the nervous and endocrine systems coordinate digestion. We then explore the cephalic phase, which initiates digestion before food enters the stomach. Next, the gastric phase covers the stomach's role in chemical digestion and protein breakdown. Finally, the intestinal phase explains nutrient absorption in the small intestine and waste concentration in the large intestine. By the end of this chapter, you will have a deeper understanding of how the digestive system works to support overall health. In this section, we will explore the anatomy of the digestive system, a complex network of organs that work together to break down food and absorb nutrients. The digestive system consists of two main components, the oral cavity and the gastrointestinal tract. Digestion begins in the oral cavity, which includes the mouth and pharynx. Here, food is mechanically broken down by chewing and chemically processed by enzymes found in saliva. From there, food travels into the gastrointestinal tract, which includes the esophagus, stomach, small intestine, and large intestine. The digestive tract refers to the section of the gastrointestinal tract that extends from the stomach to the anus. It plays a primary role in digestion, involving both chemical and mechanical processes that break down food into absorbable nutrients. To regulate the movement of food and prevent backflow, the gastrointestinal tract is divided by muscular valves called sphincters, which act as one-way valves controlling the passage of food between different sections of the digestive system. Understanding the anatomy of the digestive system is essential to comprehending how our body efficiently processes and absorbs nutrients. The digestive system is essentially a long, hollow tube that extends from the mouth to the anus. It plays a crucial role in breaking down food, absorbing nutrients, and eliminating waste. Several accessory glandular organs assist in digestion by secreting essential substances. The salivary glands, pancreas, and liver all contribute enzymes and other secretions that aid in the breakdown of food. As food moves through the digestive tract, it mixes with digestive secretions, forming a semi-liquid substance called chyme. This mixture is essential for the breakdown and absorption of nutrients. The products of digestion must be absorbed into the body to provide energy and sustain vital functions. First, nutrients pass across the epithelial lining of the digestive tract into the interstitial fluid. From there, they enter either the bloodstream or the lymphatic system, allowing them to be transported throughout the body. Once digestion is complete, waste materials, including undigested food and metabolic byproducts, are eliminated. These materials continue moving through the digestive tract until they are excreted through the anus. Because the digestive system is continuously exposed to the external environment, it also serves as a habitat for commensal microorganisms. These beneficial bacteria play a key role in digestion, immunity, and overall gut health. By understanding the structure and function of the digestive system, we gain insight into how our bodies efficiently process food and maintain balance. The digestive system is a continuous tube that processes food from ingestion to elimination, ensuring the body absorbs essential nutrients while removing waste. Digestion begins in the oral cavity, where food is broken down by chewing and mixed with saliva, which contains digestive enzymes secreted by the salivary glands. This process starts the breakdown of carbohydrates and prepares food for further digestion. Once swallowed, food travels through the esophagus, a muscular tube that uses coordinated contractions known as peristalsis to move food toward the stomach. The stomach is divided into three main regions: the fundus, body, and antrum. Here, food is mixed with gastric juices, including hydrochloric acid and digestive enzymes, which further break down proteins. The pylorus, controlled by the pyloric valve, regulates the passage of partially digested food into the small intestine. The small intestine is the primary site of nutrient absorption and consists of three sections: the duodenum, jejunum, and ileum. In this region, digestive enzymes from the pancreas and bile from the liver aid in the digestion of fats, proteins, and carbohydrates. Following nutrient absorption, any remaining waste enters the large intestine, where water is reabsorbed and beneficial bacteria further process the material. The large intestine consists of the colon, rectum, and anus, with the external anal sphincter controlling the final elimination of solid waste, known as feces. This figure provides a detailed look at the digestive system, including an overview of its major structures, a closer look at the salivary glands, and a detailed view of the stomach. In the first image, we see the digestive system as a whole. Food enters through the oral cavity, where digestion begins with chewing and the secretion of enzymes from the salivary glands. From there, food travels down the esophagus into the stomach, where it is further broken down. The liver, gallbladder, pancreas, small intestine, and large intestine all contribute to digestion and nutrient absorption. Waste materials then pass through the rectum before being excreted. The second image focuses on the salivary glands, which play a crucial role in the initial breakdown of food. There are three main salivary glands: the parotid gland, located near the cheeks, the submandibular gland, found beneath the lower jaw, and the sublingual gland, located under the tongue. These glands produce saliva, which contains enzymes that begin digesting carbohydrates while also lubricating food for easier swallowing. The third image provides a detailed view of the stomach, a key organ in digestion. The stomach is divided into different regions, including the fundus, body, and antrum. Food enters through the esophagus and is mixed with gastric juices, which help break down proteins. The pyloric region, controlled by the pyloric valve, regulates the movement of food into the small intestine. The inner surface of the stomach contains rugae, or folds, which allow the stomach to expand and accommodate food. This figure provides a detailed view of the digestive system, focusing on the small intestine and a sectional view of the stomach. In the first image, we see the structure of the small intestine. The small intestine is responsible for most nutrient absorption and is composed of three main regions: the duodenum, jejunum, and ileum. The inner surface of the small intestine is lined with circular folds, which increase the surface area for absorption. Additionally, the mesentery, a membrane that attaches the intestine to the abdominal wall, provides support and contains blood vessels, lymphatics, and nerves that aid digestion and absorption. The small intestine also contains villi, small finger-like projections that further maximize nutrient absorption. Each villus is lined with epithelial cells and contains a lacteal, which is part of the lymphatic system and plays a role in absorbing fats. The second image presents a sectional view of the stomach, showcasing its complex layers and specialized structures. The stomach's mucosa is lined with gastric pits, which open into gastric glands that secrete digestive enzymes and hydrochloric acid. These secretions help break down food into a semi-liquid substance known as chyme. The muscular layers of the stomach, including the circular, longitudinal, and oblique muscles, work together to churn food and mix it with gastric juices. Below the muscular layers, the enteric plexus is a network of nerves that coordinate stomach contractions to propel food into the small intestine. Together, the small intestine and stomach play a vital role in digestion, ensuring that nutrients are efficiently absorbed while preparing waste for elimination. The wall of the gastrointestinal tract is composed of 4 distinct layers, each with specialized functions that contribute to digestion and absorption. These layers include structural modifications that enhance the efficiency of nutrient processing. The first layer is the mucosa, which forms the inner lining of the digestive tract. It consists of 3 sublayers. The mucosal epithelium provides a protective barrier and is responsible for the secretion of digestive enzymes and mucus. Beneath it, the lamina propria, a connective tissue layer, contains immune components such as Peyer's patches, which are part of the gut-associated lymphoid tissue and help defend against pathogens. The third sublayer, the muscularis mucosae, consists of thin, smooth muscle fibers that aid in the movement of food and secretions within the digestive tract. The second layer, known as the submucosa, is composed of connective tissue that provides structural support. This layer contains the submucosal plexus, a network of nerves that is part of the enteric nervous system. This plexus plays a crucial role in regulating glandular secretions and blood flow within the digestive tract. To maximize digestion and absorption, the gastrointestinal tract contains several modifications that increase surface area. Structures such as rugae and plicae allow the stomach and intestines to expand. Villi and microscopic projections known as microvilli significantly enhance the surface area for nutrient absorption. Additionally, specialized glands, crypts, and submucosal glands contribute to secretion and immune defense within the digestive tract. Together, these layers ensure the efficient breakdown, absorption, and movement of food through the digestive system. The gastrointestinal tract is composed of 4 distinct layers, each playing a crucial role in digestion, absorption, and movement of food. In addition to the mucosa and submucosa, the outer layers include the muscularis externa and the serosa. The submucosa is the middle layer of the gastrointestinal wall and consists of connective tissue that provides structural support and elasticity. This layer also houses the submucosal plexus, a part of the enteric nervous system responsible for regulating glandular secretions and local blood flow. Surrounding the submucosa is the muscularis externa, which forms the outer wall of the gastrointestinal tract. It is composed of 2 layers of smooth muscle that coordinate rhythmic contractions, known as peristalsis, to propel food along the digestive tract. Sandwiched between these muscle layers is the enteric plexus, another component of the enteric nervous system that regulates muscle contractions and motility. The serosa is the outermost covering of the digestive tract and is a continuation of the peritoneal membrane. This thin layer forms protective sheets of mesentery, which help anchor the digestive organs in place while allowing movement and reducing friction between them. Together, these layers ensure that food is efficiently broken down, nutrients are absorbed, and waste is moved through the digestive tract. The structural complexity of the gastrointestinal wall highlights the intricate coordination required for proper digestion and overall gut function. This figure presents a sectional view of the small intestine, highlighting its structural adaptations for efficient digestion and absorption. The small intestine is lined with villi, which are finger-like projections that significantly increase the surface area for nutrient absorption. Between these villi are crypts, which contain specialized cells that help in secretion and cell renewal. The mucosa, the innermost layer, contains epithelial cells that facilitate nutrient absorption. Beneath it lies the muscularis mucosae, a thin layer of smooth muscle that helps maintain movement within the intestinal wall. The submucosa houses blood vessels, lymphatic vessels, and nerve networks essential for digestion and absorption. Notably, Peyer's patches, which are part of the gut-associated lymphoid tissue, play a critical role in immune defense, protecting the intestines from harmful pathogens. The submucosal plexus, a network of nerves, helps regulate secretion and blood flow in the digestive tract. The muscularis externa consists of two layers of smooth muscle, an inner circular muscle layer and an outer longitudinal muscle layer. These muscles work together to generate peristalsis, the wave-like contractions that propel food through the digestive system. The enteric plexus, located between these muscle layers, controls the coordination of these contractions. The serosa, the outermost layer, provides structural support and helps anchor the small intestine within the abdominal cavity. Through these specialized structures, the small intestine efficiently absorbs nutrients while maintaining a balance between digestion and immune protection. The digestive system carries out 4 essential processes that ensure the body effectively breaks down food, absorbs nutrients, and eliminates waste. These processes include digestion, absorption, secretion, and motility. The first process, digestion, involves the breakdown of food into smaller molecules. This occurs through mechanical digestion, which includes chewing and muscular contractions, and chemical digestion, which involves enzymes and acids that break down complex molecules into absorbable forms. The second process, absorption, occurs primarily in the small intestine. During this phase, nutrients such as carbohydrates, proteins, fats, vitamins, and minerals pass through the intestinal lining into the bloodstream or lymphatic system, where they are transported to cells throughout the body. The third process, secretion, involves the release of digestive enzymes, hormones, mucus, and other substances that facilitate digestion. Organs such as the salivary glands, stomach, pancreas, and intestines produce these secretions, which aid in breaking down food, protecting the digestive tract, and regulating digestion. The fourth and final process, motility, refers to the movement of food and waste through the digestive system. This is achieved through coordinated muscular contractions, including peristalsis, which propels food forward, and segmentation, which mixes contents to enhance digestion and absorption. Together, these 4 processes ensure that nutrients are efficiently extracted from food, waste is properly eliminated, and the body maintains its overall metabolic balance. This figure illustrates the 4 fundamental processes of the digestive system: digestion, absorption, secretion, and motility. These processes work together to break down food, extract essential nutrients, and transport them throughout the body. Digestion is the first step in processing food. It involves both mechanical breakdown, such as chewing and churning in the stomach, and chemical digestion, where enzymes break down complex food molecules into smaller absorbable units. This prepares nutrients for absorption. Once food is digested, the next step is absorption. This process allows nutrients to pass from the lumen of the digestive tract into the interstitial fluid and bloodstream. Carbohydrates, proteins, fats, vitamins, and minerals are absorbed primarily in the small intestine, where they enter either the circulatory or lymphatic system for distribution to cells. Secretion plays a crucial role in digestion by facilitating the breakdown and movement of food. Digestive enzymes, mucus, acids, and hormones are secreted by various organs, including the salivary glands, stomach, pancreas, and intestines. These secretions help regulate digestion, protect the gut lining, and enhance nutrient breakdown. Finally, motility ensures that food and waste move efficiently through the digestive tract. Peristalsis, a series of wave-like muscular contractions, propels food forward, while segmentation movements mix food with digestive secretions, optimizing absorption. These coordinated contractions allow for efficient digestion and elimination of waste. By working together, these four processes enable the body to extract nutrients, regulate digestion, and maintain overall health. Motility is a fundamental function of the digestive system, ensuring the movement and mixing of food throughout the gastrointestinal tract. The smooth muscles of the digestive tract contract spontaneously, performing two essential tasks. The first task is propelling food through the digestive tract, allowing it to move efficiently from the mouth to the anus. The second is mechanically mixing food with digestive enzymes and secretions, breaking it down into uniformly small particles for optimal digestion and absorption. Different regions of the gastrointestinal tract exhibit distinct types of contractions. Tonic contractions occur in certain sphincters and regions of the digestive tract, lasting for minutes to hours. These contractions help maintain muscle tone and regulate the passage of food. Phasic contractions, on the other hand, occur in shorter bursts, lasting only seconds, and are responsible for mixing and propelling food at different stages of digestion. The rhythmic contraction of these smooth muscles is coordinated by slow-wave potentials, which originate from specialized pacemaker cells known as the interstitial cells of Kajal. These cells generate electrical impulses that regulate the contraction patterns of the digestive tract, ensuring a controlled and efficient process. Through these coordinated contractions, the digestive system efficiently moves and processes food, maintaining the balance between digestion, absorption, and waste elimination. The smooth muscle of the gastrointestinal tract exhibits three distinct patterns of contraction, each serving a specific role in digestion and movement of food. The first pattern is the migrating motor complex. This occurs between meals, when the stomach and small intestine are relatively empty. It is a series of rhythmic contractions that sweep undigested material and bacteria through the digestive tract, helping to maintain cleanliness and prevent bacterial overgrowth. The second pattern is peristalsis, a wave-like motion that propels food forward through the digestive tract. As food, or a bolus, moves through the esophagus, stomach, and intestines, coordinated contractions push it along, ensuring continuous digestion and absorption. The third pattern is segmental contractions, which occur primarily in the small and large intestines. These contractions mix food with digestive enzymes and secretions, promoting efficient nutrient absorption. Instead of moving food forward, segmental contractions churn and break it down into smaller particles while increasing contact time with the intestinal walls. Each of these contraction patterns plays a critical role in digestion, absorption, and waste removal, ensuring that the gastrointestinal system functions efficiently. This figure highlights key aspects of gastrointestinal motility, focusing on slow waves and the migrating motor complex. These mechanisms play a vital role in regulating the movement of food and waste through the digestive tract. In the first image, we see a graphical representation of slow waves, which are spontaneous depolarizations in gastrointestinal smooth muscle. Unlike typical nerve impulses, slow waves do not always trigger muscle contractions. Only when the membrane potential surpasses the threshold do action potentials occur, leading to smooth muscle contractions. The force and duration of muscle contraction are directly related to the amplitude and frequency of these action potentials. This allows for controlled, rhythmic contractions that facilitate digestion and absorption. The second image illustrates the migrating motor complex, a specialized contraction pattern that occurs between meals when the digestive tract is empty. This complex begins in the stomach and moves through the small intestine, eventually reaching the large intestine. It serves as a housekeeping mechanism, clearing undigested food particles and bacteria from the digestive tract. The migrating motor complex helps prevent bacterial overgrowth and prepares the intestines for the next meal. By regulating contractions through slow waves and the migrating motor complex, the gastrointestinal system ensures efficient digestion, absorption, and waste elimination. This figure highlights two key types of gastrointestinal motility: peristaltic contractions, responsible for forward movement of food, and segmental contractions, which are essential for mixing. In the first image, we see peristalsis, a wave-like motion that propels food, or the bolus, through the digestive tract. This process occurs as smooth muscle contractions push the bolus forward while the segment ahead relaxes, allowing for continuous movement. Peristalsis is crucial for moving food through the esophagus, stomach, small intestine, and large intestine, ensuring digestion and absorption continue efficiently. In the second image, segmental contractions are shown. Unlike peristalsis, segmental contractions do not move food forward, but instead function to mix food with digestive enzymes and secretions. This occurs through alternating contractions of different segments of the intestine, allowing for more effective digestion and nutrient absorption. The mixing action also helps expose food particles to the intestinal walls, where absorption occurs. Both peristalsis and segmental contractions are essential for digestive function. Peristalsis ensures the efficient movement of food and waste, while segmental contractions maximize digestion and absorption. The regulation of gastrointestinal function is controlled by a complex network of neural pathways, including the enteric nervous system and the central nervous system. The enteric nervous system, often referred to as the second brain, can function independently of the central nervous system. It plays a critical role in controlling motility, secretion, and the growth of the digestive system. The enteric nervous system shares several key features with the central nervous system. It contains intrinsic neurons that function within the digestive tract, as well as extrinsic neurons, which connect the central nervous system to the digestive system. Additionally, it utilizes neurotransmitters and neuromodulators to regulate digestive processes, relies on glial support cells similar to those found in the brain, and maintains a diffusion barrier that protects its environment. The enteric nervous system also acts as an integrating center, allowing it to process sensory information and generate appropriate responses without direct input from the brain. Gastrointestinal regulation occurs through short and long reflexes. Short reflexes are processed entirely within the enteric nervous system, allowing for localized and rapid responses to changes in the digestive environment. In contrast, long reflexes involve the central nervous system and can influence digestion from outside the gastrointestinal tract. One example is the cephalic reflex, which originates outside the digestive system. This reflex is triggered by the sight, smell, or thought of food and prepares the digestive tract for incoming nutrients by stimulating secretions and motility. Together, these regulatory systems ensure precise control of digestive processes, adapting to the body's needs and maintaining overall digestive health. The digestive system relies on a variety of gastrointestinal hormones to regulate digestion, nutrient absorption, and energy balance. These hormones are secreted by specialized cells in response to specific stimuli and target various organs to coordinate digestive function. The stomach secretes gastrin, which is produced by gastrin secreting cells in response to peptides, amino acids, and neural reflexes. Gastrin stimulates gastric acid secretion and mucosal growth, preparing the stomach for digestion. However, its release is inhibited by somatostatin, which prevents excessive acid production. In the intestine, several hormones play key roles in digestion. Cholecystokinin is released in response to fatty acids and amino acids. It stimulates gallbladder contraction, promoting the release of bile for fat digestion, and enhances pancreatic enzyme secretion. Cholecystokinin also inhibits gastric emptying and acid secretion, ensuring that digestion occurs at an optimal pace. Additionally, it plays a role in promoting the sensation of fullness, reducing the desire to eat. Secretin is another hormone released when acid enters the small intestine. Its primary function is to stimulate the pancreas to secrete bicarbonate, which neutralizes stomach acid to protect the intestinal lining. Like cholecystokinin, secretin also inhibits gastric emptying and acid secretion to regulate the flow of digestion. Motilin is responsible for stimulating the migrating motor complex, which is a series of contractions that sweep undigested material through the digestive tract. It is released in fasting periods every 1.5 to 2 hours, but is inhibited once a meal is consumed. The small intestine also releases hormones that regulate insulin and glucose balance. Gastric inhibitory peptide and glucagon-like peptide 1 respond to glucose, fatty acids, and amino acids. Both hormones stimulate insulin release from the pancreas, helping to regulate blood sugar levels. Additionally, they inhibit glucagon secretion and gastric function, further slowing digestion to allow for efficient nutrient absorption. Glucagon-like peptide 1 also promotes the sensation of fullness, reducing food intake. Together, these gastrointestinal hormones ensure that digestion, absorption, and energy balance are tightly regulated, allowing the body to process food efficiently while maintaining overall stability. This figure provides an overview of digestive function, highlighting the roles of different regions of the digestive system in secretion, digestion, absorption, and motility. Digestion begins in the oral cavity and esophagus. The salivary glands secrete saliva, which contains enzymes that initiate the breakdown of carbohydrates. Although no absorption occurs here, chewing and swallowing facilitate the movement of food into the esophagus. In the stomach, digestion becomes more complex. Specialized cells in the stomach lining secrete hydrochloric acid, digestive enzymes such as pepsinogen and gastric lipase, and protective mucus. These secretions help break down proteins and fats while also acting as a barrier against harmful microorganisms. The stomach also absorbs lipid-soluble substances, such as alcohol and aspirin. Peristaltic contractions mix and propel food into the small intestine for further digestion. The small intestine is the primary site of digestion and nutrient absorption. It secretes enzymes, mucus, and hormones, including cholecystokinin, secretin, and gastric inhibitory peptide. These hormones regulate digestive function by stimulating the release of bicarbonate from the pancreas, bile from the liver, and digestive enzymes from the pancreas. The small intestine digests polypeptides, carbohydrates, fats, and nucleic acids, absorbing essential nutrients such as amino acids, monosaccharides, fatty acids, and water-soluble vitamins. Mixing and propulsion occur primarily through segmental contractions, allowing for efficient nutrient absorption. The large intestine primarily functions in water and ion absorption while forming and storing waste. The secretion of mucus helps lubricate the passage of undigested material. The large intestine absorbs water, minerals, ions, and vitamins, while its resident bacteria contribute to the production of certain nutrients. Segmental contractions mix the contents, while mass movement contractions propel waste toward the rectum for elimination. Through the coordinated efforts of these digestive organs, the body efficiently processes food, absorbs essential nutrients, and eliminates waste, ensuring overall health and metabolic balance. The cephalic phase of digestion begins before food even enters the mouth. This phase is triggered by the sight, smell, or thought of food, activating a reflex that starts in the brain. When a person anticipates food, or when food is placed in the oral cavity, neurons in the medulla oblongata of the brainstem are activated. This neural response initiates the cephalic phase, preparing the digestive system for the intake and processing of food. Chemical and mechanical digestion both begin in the mouth. Salivary glands play a crucial role in this phase, releasing saliva, which serves four key functions. First, saliva softens and lubricates food, making it easier to swallow. Second, it contains enzymes that chemically digest starch, beginning the breakdown of complex carbohydrates. Third, saliva enhances the sense of taste, helping the body evaluate the food being consumed. Lastly, saliva provides a defensive function, containing antimicrobial compounds that help protect against pathogens. Mechanical digestion begins with chewing, also known as mastication. This process physically breaks food into smaller particles, increasing its surface area for enzyme activity and making it easier to swallow. The cephalic phase plays a vital role in priming the digestive system, ensuring that the stomach and intestines are prepared for efficient digestion and absorption once food enters the body. Saliva plays a crucial role in digestion and is classified as an exocrine secretion because it is released by glands with ducts directly into the oral cavity. There are three pairs of major salivary glands, each with distinct functions. The parotid glands, located near the cheeks, produce a watery solution rich in digestive enzymes. These enzymes, such as amylase, begin the breakdown of carbohydrates. The sublingual glands, found beneath the tongue, secrete mucus-rich saliva. This thicker secretion helps lubricate food, making swallowing easier. The submandibular glands, positioned beneath the lower jaw, produce a mixture of both watery enzyme-rich saliva and mucus, combining the functions of lubrication and enzymatic digestion. Salivary secretion is regulated by the autonomic nervous system, meaning it occurs involuntarily. Stimulation of the parasympathetic nervous system increases saliva production, such as when food is present in the mouth or when a person anticipates eating. In contrast, sympathetic activation can reduce saliva flow, often causing a dry mouth during stress or anxiety. Saliva not only aids in digestion, but also plays an important role in taste, swallowing, and protection against pathogens, ensuring a well-functioning oral and digestive system. The process of swallowing, known as deglutition, is a highly coordinated reflex that ensures food moves safely from the mouth to the stomach while protecting the airway. This reflex is controlled by the medulla oblongata, which integrates sensory input and motor responses. Swallowing occurs in three distinct phases. In the oral phase, food is prepared for swallowing. The tongue pushes the bolus of food against the soft palate and toward the back of the mouth, triggering the swallowing reflex. The soft palate elevates, closing off the nasal passages to prevent food from entering the nasal cavity. The pharyngeal phase begins as the bolus moves toward the throat. During this stage, swallowing is momentarily inhibited to ensure food does not enter the airway. The epiglottis folds downward, covering the entrance to the trachea, while the larynx moves upward and forward to assist in airway protection. The upper esophageal sphincter relaxes, allowing food to pass from the pharynx into the esophagus. In the esophageal phase, the bolus moves downward through the esophagus, propelled by peristaltic contractions. These wave-like muscular movements push food toward the stomach, while gravity assists in its descent. The lower esophageal sphincter relaxes, allowing food to enter the stomach for digestion. This intricate reflex ensures that food is efficiently transported while safeguarding the respiratory system. Swallowing is an essential function that allows for safe and efficient digestion. The gastric phase of digestion begins when food enters the stomach, triggering a series of neural and chemical responses that facilitate digestion. The stomach plays three essential roles in this phase: storage, digestion, and defense. The first function is storage. The upper region of the stomach temporarily holds food, allowing it to be processed gradually. This prevents the small intestine from becoming overwhelmed and ensures controlled digestion. The second function is digestion, which occurs primarily in the lower region of the stomach. Here, lipids and proteins are broken down by gastric acid, digestive enzymes, and signaling molecules. Hydrochloric acid creates an acidic environment that denatures proteins and activates enzymes such as pepsin, which breaks proteins into smaller peptides. Other hormones and paracrine molecules regulate gastric activity and coordinate digestion with the rest of the digestive tract. The third function of the stomach is defense. The acidic environment plays a critical role in protecting the body from ingested pathogens by killing harmful bacteria and microorganisms before they can enter the intestines. Digestive activity in the stomach is controlled by reflexes that begin in the brain and continue in the stomach. The cephalic phase, initiated by the sight, smell, or thought of food, triggers a long reflex through the vagus nerve, preparing the stomach for digestion. Once food enters the stomach, it initiates short reflexes that regulate acid secretion, enzyme release, and muscle contractions. By integrating storage, digestion, and defense, the stomach efficiently processes food, preparing it for further digestion and absorption in the small intestine. The cephalic and gastric phase reflexes play an essential role in regulating digestion by preparing the stomach for food intake and coordinating its response once food arrives. These reflexes involve the nervous system, chemical messengers, and muscle contractions to ensure efficient digestion. The cephalic phase begins when the sight, smell, or thought of food activates sensory receptors in the brain. This information is processed in the medulla oblongata, which sends signals through the vagus nerve to the stomach. This long vagal reflex stimulates gastric secretions and motility, priming the stomach for digestion before food even enters. Once food reaches the stomach, the gastric phase begins. The stretching of the stomach walls, along with the presence of peptides and amino acids, triggers short reflexes that further stimulate digestion. These short reflexes involve sensory receptors in the stomach lining, which send signals to the enteric nervous system. In response, target cells in the gastric mucosa release digestive enzymes, hydrochloric acid, and hormones that regulate digestion. These reflexes also play a role in defense, as the acidic environment helps kill harmful microorganisms. The coordination between the long vagal reflex and short gastric reflexes ensures that digestion occurs in a controlled and efficient manner. By integrating these neural and hormonal pathways, the body optimizes digestion, ensuring food is broken down properly and nutrients are efficiently absorbed. The stomach plays a crucial role in digestion by secreting various substances that break down food while also protecting the digestive system. These secretions come from gastric glands, which are found in deep folds known as gastric pits within the stomach lining. One of the key hormones secreted is gastrin, which is produced by gastrin-secreting cells. The release of gastrin is stimulated by gastrin-releasing peptide, a signaling molecule that enhances digestive activity. Acid secretion is carried out by parietal cells, which produce hydrochloric acid. This strong acid denatures proteins, activates digestive enzymes and kills harmful microorganisms that enter the stomach with food. The stomach also secretes digestive enzymes, which further break down food. Chief cells produce pepsinogen, an inactive enzyme that is converted into pepsin in the presence of acid. Pepsin plays a crucial role in protein digestion. Additionally, chief cells secrete gastric lipase, which aids in the breakdown of fats. In addition to enzymes and acid, the stomach also releases paracrine signals, which help regulate digestion. Enterochromaffin-like cells secrete histamine, which stimulates acid secretion. Parietal cells also produce intrinsic factor, a protein necessary for the absorption of vitamin B12 in the small intestine. Meanwhile, these cells release somatostatin, a hormone that inhibits gastric acid secretion to prevent excessive acidity. By combining hormonal, enzymatic, and paracrine secretions, the stomach efficiently processes food while maintaining a delicate balance between digestion and protection. The stomach plays a dual role in digestion, breaking down food while protecting itself from its own harsh environment. To maintain this balance, the stomach relies on a mucus bicarbonate barrier that shields the stomach lining and prevents self-digestion. The stomach lining is coated with mucus, which acts as a physical barrier between the highly acidic gastric juices and the stomach wall. This prevents the acid from damaging the tissue and helps maintain a healthy digestive environment. Additionally, the stomach contains bicarbonate, a substance that neutralizes acid near the stomach lining, creating a protective alkaline layer. This buffering action further prevents damage to the stomach's epithelial cells. The mucus cells are responsible for producing both mucus and bicarbonate. These specialized cells are found on the luminal surface of the stomach lining and at the neck of the gastric glands. Their continuous secretion ensures that the protective barrier remains intact, even as the stomach secretes strong acids and digestive enzymes. By maintaining this delicate balance, the stomach is able to efficiently digest food while protecting itself from its own corrosive environment, ensuring both digestion and defense function in harmony. The stomach lining, also known as the gastric mucosa, contains specialized secretory cells that produce substances essential for digestion and protection. These secretions include mucus, acid, enzymes, and hormones, each with a specific function in regulating gastric activity. The mucus surface cells secrete mucus, which forms a physical barrier between the stomach lining and the highly acidic gastric environment. This prevents damage to the stomach epithelium. The mucus neck cells secrete bicarbonate, which buffers gastric acid and further protects the stomach lining by neutralizing acid near the epithelium. Parietal cells are responsible for secreting hydrochloric acid, which activates pepsin, a digestive enzyme, and kills harmful bacteria that may enter the digestive tract. Additionally, parietal cells release intrinsic factor, a protein necessary for the absorption of vitamin B12 in the small intestine. The release of these substances is stimulated by acetylcholine, gastrin, and histamine. The enterochromaffin-like cells produce histamine, a molecule that stimulates acid secretion, further enhancing digestion. These cells are activated by acetylcholine and gastrin, which are released in response to food intake. Chief cells produce pepsinogen, an inactive enzyme that is converted into pepsin in the presence of acid. Pepsin is crucial for breaking down proteins into smaller peptides. Chief cells also secrete gastric lipase, which aids in the digestion of fats. The release of these enzymes is stimulated by acetylcholine and acid secretion. To regulate acid production, these cells secrete somatostatin, a hormone that inhibits gastric acid secretion when acid levels in the stomach become too high. Finally, G cells secrete gastrin, a hormone that stimulates acid production. Gastrin is released in response to acetylcholine, peptides, and amino acids, signaling the stomach to increase digestive activity. By coordinating these secretions, the stomach ensures that food is efficiently digested while maintaining a protective balance to prevent damage to the stomach lining. The stomach secretes powerful acids to aid digestion, but it must also protect itself from self-digestion. This balance is achieved through the mucous bicarbonate barrier and the regulated secretion of gastric acid. The first image illustrates the mucous bicarbonate barrier, which serves as both a physical and chemical defense against the harsh gastric environment. The stomach's gastric juice has a very low pH, around 2, making it highly acidic. To protect the stomach lining, mucous cells secrete a thick mucus layer, which acts as a physical barrier between the stomach epithelium and gastric acid. Additionally, bicarbonate ions are secreted into this mucus layer, creating a chemical barrier that neutralizes acid near the stomach lining. As a result, while the stomach lumen remains highly acidic, the cell surface maintains a near neutral pH of around 7, preventing damage to the stomach tissue. The second image focuses on the mechanism of acid secretion in the stomach, which is carried out by parietal cells. Within parietal cells, carbon dioxide combines with water to form carbonic acid, which then dissociates into hydrogen ions and bicarbonate ions. The hydrogen ions are actively transported into the stomach lumen by a hydrogen-potassium pump, which uses energy in the form of adenosine triphosphate to exchange hydrogen ions for potassium ions. Meanwhile, chloride ions move into the stomach lumen, combining with hydrogen ions to form hydrochloric acid, the key component of gastric juice. At the same time, bicarbonate ions are transported into the bloodstream, creating the alkaline tide, a temporary increase in blood pH that occurs after a meal. By coordinating mucus and bicarbonate secretion with acid production, the stomach ensures efficient digestion while maintaining a protective balance to prevent damage to its own tissues. The intestinal phase of digestion is the final stage of food processing, where nutrients are absorbed, digestion is completed, and motility is carefully regulated to ensure efficiency. Motility in the small intestine is controlled by segmental and peristaltic contractions. Segmental contractions mix the partially digested food, or chyme, with digestive enzymes, allowing for thorough breakdown and absorption. Peristaltic contractions propel chyme forward, ensuring that the small intestine is not overwhelmed with excessive material at once. This controlled movement ensures that digestion is completed and nutrients have ample time to be absorbed. The small intestine maximizes surface area to enhance nutrient absorption. This is achieved through villi and crypts, which are finger-like and pocket-like structures that increase contact with digested food. Additionally, the brush border, composed of microvilli on the intestinal epithelial cells, further expands the surface area, facilitating the efficient absorption of nutrients. Once nutrients are absorbed by the intestinal epithelium, they are distributed throughout the body. Most nutrients enter the circulatory system through the hepatic portal system, which directs absorbed substances to the liver for processing before they enter the general bloodstream. However, fats follow a different pathway, entering the lymphatic circulation before eventually merging with the circulatory system. Through precise regulation of motility, surface area expansion, and specialized nutrient absorption pathways, the intestinal phase ensures that the body efficiently extracts and distributes essential nutrients while maintaining digestive balance. The small intestine is the primary site of nutrient absorption, and its efficiency is enhanced by villi and crypts, which significantly increase the surface area for digestion and absorption. Each villus is covered by specialized cells known as enterocytes, which transport nutrients and ions from the digestive tract into the bloodstream. The surface of these cells features microvilli, forming the brush border, which further increases absorption capacity and contains enzymes that aid in digestion. Beneath the epithelial layer, capillaries transport most absorbed nutrients, such as carbohydrates and amino acids, into the circulatory system. Meanwhile, lacteals, which are lymphatic vessels, absorb dietary fats and transport them through the lymphatic system before they enter the bloodstream. The crypts, located at the base of the villi, serve an essential role in maintaining intestinal function. Stem cells in the crypts divide to replace damaged epithelial cells, ensuring the continuous renewal of the intestinal lining. Crypts also contain goblet cells, which secrete mucus to protect the lining and facilitate the movement of digested food. Additionally, crypt cells release ions and water, contributing to fluid balance in the intestine. Endocrine cells found in the crypts secrete hormones that regulate digestion and absorption, while the muscularis mucosae, a thin layer of smooth muscle, helps move nutrients from the villi into the circulation. Through the combined action of villi, enterocytes, capillaries, lacteals, and crypts, the small intestine ensures that the body efficiently absorbs and distributes essential nutrients while maintaining intestinal health. The hepatic portal system plays a vital role in processing nutrients absorbed from the digestive tract while also protecting the body from harmful substances. This system acts as a biological filter, ensuring that absorbed nutrients are processed by the liver before entering the general circulation. When nutrients are absorbed in the small intestine, they do not immediately enter the systemic bloodstream. Instead, they are transported through capillaries in the digestive tract, which collect blood from the stomach, intestines, pancreas, and spleen. This nutrient-rich blood flows into the hepatic portal vein, which carries it directly to the liver. Within the liver, capillaries further filter and process the blood, allowing nutrients to be metabolized, stored, or released as needed. The liver also detoxifies potentially harmful substances, including xenobiotics, compounds foreign to the body, such as drugs and toxins, before they can enter general circulation. After filtration, the processed blood exits the liver through the hepatic vein and joins the inferior vena cava, which carries it to the heart for distribution to the rest of the body. Meanwhile, oxygen-rich blood from the hepatic artery supplies the liver with the necessary energy to perform its functions. This system ensures that nutrients are properly utilized and harmful substances are neutralized, maintaining metabolic balance and protecting overall health. The small intestine plays a crucial role in digestion by secreting substances that aid in the breakdown and absorption of nutrients. These intestinal secretions include digestive enzymes, bile, bicarbonate, mucus, and isotonic sodium chloride solution, all of which contribute to the efficiency of digestion. The first type of secretion is digestive enzymes, which are produced by the pancreas and enterocytes in the small intestine. These enzymes break down carbohydrates, proteins, and fats, converting them into smaller molecules that can be absorbed by intestinal cells. The second secretion is bile, which is produced by the liver and stored in the gallbladder. Bile contains bile salts, which emulsify fats, breaking them into smaller droplets that can be easily digested by lipase enzymes. The third secretion is bicarbonate, which is released primarily by the pancreas. Bicarbonate neutralizes stomach acid as chyme enters the small intestine, creating an optimal pH environment for digestive enzymes to function effectively. The fourth secretion is mucus, which is produced by goblet cells in the intestinal lining. Mucus lubricates and protects the intestinal walls, preventing damage from digestive enzymes and the movement of food through the tract. The fifth and final secretion is isotonic sodium chloride solution, which helps maintain the fluid balance in the intestine and facilitates the movement of digested material. This secretion is regulated by the cystic fibrosis transmembrane conductance regulator channel, which controls the transport of chloride ions across the intestinal epithelium. This movement of chloride ions drives the secretion of sodium and water, ensuring that digestive contents remain fluid and easily transported. Together, these secretions create an environment that optimizes digestion and absorption, allowing the body to efficiently process food while maintaining intestinal health. The secretion of isotonic sodium chloride solution plays an essential role in maintaining fluid balance in the intestinal tract. This process ensures that digestive contents remain hydrated, aiding in nutrient absorption and the movement of material through the intestine. This figure illustrates the mechanism of sodium chloride secretion, which occurs in intestinal and colonic crypt cells as well as salivary gland acini cells. The process begins when sodium, potassium, and chloride ions enter the intestinal cells from the interstitial fluid via a cotransporter protein. This step requires energy from adenosine triphosphate to drive the movement of ions against their concentration gradient. Next, chloride ions move into the lumen of the intestine through a specialized channel known as the cystic fibrosis transmembrane conductance regulator channel. This channel is critical for the regulation of chloride ion transport, and its dysfunction is associated with cystic fibrosis, a condition that leads to thickened mucus secretions. As chloride ions accumulate in the intestinal lumen, they create an electrical gradient that attracts sodium ions. These sodium ions follow via a pericellular pathway, moving between the intestinal cells rather than through them. Finally, water follows sodium and chloride ions into the lumen, ensuring that the intestinal environment remains hydrated and facilitating the movement of digested food through the digestive tract. This process of isotonic sodium chloride secretion is essential for maintaining proper digestive function, fluid balance, and overall intestinal health. The pancreas is a vital organ that serves both endocrine and exocrine functions, playing a key role in digestion and metabolism. The endocrine portion of the pancreas, known as the islets of Langerhans, secretes insulin and glucagon. These hormones regulate blood glucose levels, ensuring that energy is properly stored and released as needed by the body. The exocrine portion of the pancreas is responsible for the production and secretion of digestive enzymes and bicarbonate, which are essential for breaking down food and neutralizing stomach acid. Pancreatic enzyme secretion is carried out by acinar cells, which produce inactive digestive enzymes. One of these enzymes, trypsinogen, is converted into its active form, trypsin, by brush border enteropeptidase in the small intestine. Trypsin then activates other pancreatic enzymes, enabling the digestion of proteins, fats, and carbohydrates. The pancreas also secretes bicarbonate, which is produced by duct cells. Bicarbonate plays a crucial role in neutralizing gastric acid, creating an optimal pH environment for digestive enzymes to function in the small intestine. This prevents damage to the intestinal lining and allows for efficient nutrient absorption. By integrating endocrine and exocrine functions, the pancreas ensures that the body maintains both metabolic balance and effective digestion, supporting overall health and nutrient absorption. The pancreas is a unique organ with both exocrine and endocrine functions, playing a critical role in digestion and metabolic regulation. The exocrine portion of the pancreas consists of acinar cells, which produce digestive enzymes. These enzymes break down proteins, fats, and carbohydrates, ensuring efficient digestion in the small intestine. The enzymes are secreted into the pancreatic duct, which transports them to the small intestine. Additionally, duct cells in the pancreas secrete sodium bicarbonate, which neutralizes acidic contents from the stomach. This helps maintain an optimal pH environment for enzyme activity and prevents damage to the intestinal lining. The endocrine portion of the pancreas is composed of clusters of specialized cells known as the islets of Langerhans. These cells secrete hormones directly into the bloodstream, regulating metabolism and blood sugar levels. Insulin, secreted by beta cells, lowers blood sugar levels by promoting glucose uptake into cells. Glucagon, secreted by alpha cells, increases blood sugar levels by stimulating the release of glucose from stored glycogen in the liver. The pancreatic acini form the exocrine portion, while the islet cells form the endocrine portion, allowing the pancreas to function as both a digestive and regulatory organ. By integrating digestive enzyme production, bicarbonate secretion, and hormonal regulation, the pancreas plays a central role in nutrient breakdown and metabolic homeostasis, ensuring the body maintains energy balance and efficient digestion. The pancreas plays a key role in digestion by secreting inactive enzymes and bicarbonate, both of which are essential for efficient nutrient breakdown and acid neutralization. The first image illustrates the activation of pancreatic zymogens, which are inactive enzyme precursors secreted by the pancreas. These zymogens include chymotrypsinogen, procarboxypeptidase, proelastase, and prophospholipase. When these inactive enzymes reach the small intestine, they are activated in a cascade. The brush border enzyme enteropeptidase converts trypsinogen into trypsin. Trypsin then activates the other pancreatic enzymes, allowing them to digest proteins, fats, and carbohydrates efficiently. This sequential activation process prevents premature enzyme activity, which could otherwise damage the pancreas. The second image highlights the bicarbonate secretion process, which is crucial for neutralizing acidic chyme from the stomach as it enters the small intestine. Inside pancreatic duct cells, the enzyme carbonic anhydrase catalyzes the formation of bicarbonate ions and hydrogen ions. Bicarbonate is then secreted into the lumen of the pancreas and duodenum, while hydrogen ions are transported back into the blood to maintain acid-base balance. Chloride ions play a key role in bicarbonate secretion. They enter the pancreatic cells and are transported into the lumen through the cystic fibrosis transmembrane conductance regulator channel. This movement of chloride drives the exchange of bicarbonate into the digestive tract, ensuring proper pH regulation. Additionally, sodium and water follow bicarbonate into the lumen, maintaining the fluidity of digestive secretions and facilitating enzyme function. This process ensures that the small intestine remains protected from stomach acid while creating an optimal environment for digestion. By regulating enzyme activation and bicarbonate secretion, the pancreas ensures that nutrient breakdown is efficient and that the intestinal lining remains protected, supporting overall digestive health. The liver plays a crucial role in digestion by producing bile, a substance essential for the digestion and absorption of fats. Bile is secreted by specialized liver cells called hepatocytes. It is composed of three main components: bile salts, bile pigments, and cholesterol. Bile salts emulsify fats, breaking them into smaller droplets to enhance digestion. Bile pigments, including bilirubin, are byproducts of red blood cell breakdown. Cholesterol is excreted in bile and can be used to form bile salts. Bile does not contain digestive enzymes, but instead aids in fat digestion and absorption by increasing the surface area available for lipase enzymes to act on. Once bile is produced by the liver, It is stored and concentrated in the gallbladder until needed for digestion. When dietary fats enter the small intestine, the gallbladder releases bile through the common bile duct, delivering it to the duodenum, the first segment of the small intestine. This precise regulation of bile production, storage, and release ensures that dietary fats are efficiently digested and absorbed, supporting overall digestive function. The liver is the largest internal organ in the body, weighing approximately 1.5 kilograms in an adult. It is located just under the diaphragm on the right side of the body. The liver performs many essential functions, including metabolism, detoxification, and digestion. One of its key digestive functions is the production of bile. The common hepatic duct transports bile from the liver to the gallbladder, where it is stored and concentrated until needed. When fats enter the small intestine, bile is released from the gallbladder through the common bile duct into the lumen of the small intestine. The liver receives blood from two sources. The hepatic artery delivers oxygenated blood to supply liver tissues, while the hepatic portal vein carries nutrient-rich blood absorbed from the gastrointestinal tract. This blood also contains metabolites and waste products, including hemoglobin breakdown products from the spleen. After processing, blood exits the liver through the hepatic vein. The sphincter of Oddi regulates the release of bile and pancreatic secretions into the duodenum, ensuring that bile is delivered only when needed for digestion. This coordination between the liver, gallbladder, and bile ducts allows for the efficient breakdown and absorption of dietary fats. By producing bile and regulating its release, the liver plays a crucial role in digestion and nutrient absorption while also filtering and processing substances from the blood. The liver is composed of specialized cells called hepatocytes, which are arranged in functional units known as lobules. These lobules have an irregular hexagonal structure and play a key role in processing blood and producing bile. Each lobule is centered around a central vein, which drains blood into the hepatic vein for return to circulation. Surrounding the lobule, branches of the hepatic portal vein and hepatic artery supply blood to the liver. The hepatic portal vein carries nutrient-rich blood from the digestive tract, while the hepatic artery delivers oxygenated blood to support liver function. Blood from these vessels flows through sinusoids, which are specialized capillaries that allow for the exchange of nutrients, oxygen, and waste products between hepatocytes and the bloodstream. Approximately 70% of each hepatocyte surface area faces these sinusoids, maximizing contact for efficient processing. In addition to processing blood, the liver also secretes bile. Bile is collected in small channels called bile canaliculi, which run between hepatocytes. These canaliculi merge into bile ductules, which transport bile out of the liver and into the biliary system for storage or immediate use in digestion. Through this intricate network of blood vessels, sinusoids, and bile ducts, the liver plays a central role in metabolism, detoxification, and digestion, ensuring that nutrients are processed efficiently while harmful substances are removed. The liver serves as the body's central hub for metabolism, detoxification, and nutrient processing. It receives blood from two primary sources: the hepatic portal vein, which carries nutrients, drugs, and foreign substances absorbed from the gastrointestinal tract, and the hepatic artery, which delivers oxygen-rich blood to sustain liver function. Upon entering the liver, blood is filtered and processed. The liver plays a key role in glucose and fat metabolism, protein synthesis, hormone production, urea formation, detoxification, and storage of essential nutrients. Some compounds are stored for later use, while others are broken down for excretion. Certain waste products and excess substances are eliminated through the bile duct, which transports bile salts, bilirubin, water, ions, and phospholipids to the small intestine. This secretion aids in fat digestion and waste removal. The liver also metabolizes and removes hormones, drugs, and other chemicals from circulation. By filtering bilirubin from hemoglobin breakdown and processing metabolic byproducts, the liver ensures that harmful substances are either excreted by the kidneys or detoxified for safe elimination. Blood leaving the liver through the hepatic vein contains processed nutrients, hormones, and plasma proteins, including albumin, clotting factors, and angiotensinogen. Additionally, the liver synthesizes and releases glucose, urea, vitamin D precursors, and other essential compounds to be delivered to peripheral tissues. Through these intricate metabolic and detoxification processes, the liver plays a critical role in maintaining homeostasis, ensuring that nutrients are available for the body while eliminating harmful substances. Fats, or lipids, are an essential source of energy, but because they are hydrophobic, they do not mix easily with the aqueous environment of the digestive tract. To facilitate digestion, fats must first be emulsified before enzymes can effectively break them down. The process begins when bile from the liver is released into the small intestine through the sphincter of Oddi. Bile contains bile salts, which play a crucial role in fat digestion. These bile salts have both hydrophobic and hydrophilic regions, allowing them to coat lipid droplets and form emulsions. The hydrophobic side of the bile salts associates with fat molecules, while the hydrophilic side interacts with water, breaking large fat droplets into smaller micelles. This increases the surface area available for digestion. Once emulsified, fats are acted upon by pancreatic lipase and cholipase, which are secreted by the pancreas. Lipase breaks triglycerides down into monoglycerides and free fatty acids, making them small enough to be absorbed by intestinal cells. This precise coordination between bile salts, lipase, and cholipase ensures that fats are efficiently digested and prepared for absorption, allowing the body to utilize these nutrients for energy and cell function. Fat digestion requires a complex interaction between bile salts, digestive enzymes, and specialized transport mechanisms to ensure that lipids can be broken down and absorbed efficiently. Once large fat droplets are emulsified by bile salts, they form micelles, which are small, disc-shaped structures containing bile salts, phospholipids, free fatty acids, cholesterol, monoglycerides, and diglycerides. These micelles keep fats suspended in the aqueous environment of the small intestine, allowing enzymes to access them for digestion. The breakdown of fats is carried out by lipase and cholipase, which are secreted by the pancreas. Lipase targets triglycerides, breaking them down into monoglycerides and free fatty acids, which are now small enough to be absorbed by the intestinal epithelial cells. Cholipase plays a crucial role by allowing lipase to access fat droplets despite the presence of bile salts. This precise coordination between bile salts, micelle formation, and enzymatic digestion ensures that fats are effectively processed and ready for absorption. Once inside the intestinal cells, these smaller lipid components will be repackaged into chylomicrons for transport through the lymphatic system and into circulation. Fat digestion and absorption is a multi-step process that involves bile salts, digestive enzymes, micelle formation, and specialized transport mechanisms to efficiently deliver lipids to the body. Fat digestion begins when bile salts from the liver coat fat droplets, breaking them into smaller emulsified particles. This emulsification increases the surface area, allowing pancreatic lipase and cholipase to efficiently break down triglycerides into monoglycerides and free fatty acids. These lipid components are stored inside micelles, which transport them through the aqueous environment of the small intestine. Once digestion is complete, Monoglycerides and free fatty acids diffuse out of the micelles and enter intestinal cells by simple diffusion. Cholesterol, another key lipid component, is transported into the cells through specialized mechanisms. Inside the intestinal cells, absorbed fats are reassembled. Monoglycerides and free fatty acids recombine to form triglycerides, which are then packaged with cholesterol and proteins into structures called chylomicrons within the Golgi apparatus. These chylomicrons serve as lipid transport vehicles, ensuring that fats can be distributed throughout the body. Because chylomicrons are too large to enter capillaries directly, they are absorbed into the lymphatic system through lacteals, specialized lymphatic vessels in the small intestine. From there, they travel through the lymphatic system and eventually enter the bloodstream via the vena cava, where they can be transported to tissues for energy or storage. This carefully regulated process ensures that dietary fats are efficiently broken down, absorbed, and distributed throughout the body, supporting essential cellular functions and energy production. The small intestine is the primary site of digestion and nutrient absorption. It processes fats, carbohydrates, and other macronutrients through specialized enzymes and transport mechanisms. Fat digestion begins with the emulsification of lipids by bile salts, which allows enzymes to act more efficiently. Lipases and cholipase, secreted by the pancreas, break triglycerides down into monoglycerides and free fatty acids. These molecules are then incorporated into micelles, which facilitate their absorption by intestinal cells. Once inside, they are repackaged into chylomicrons and transported through the lymphatic system for distribution to tissues. Carbohydrate digestion begins with the breakdown of complex carbohydrate polymers, such as starch and glycogen, into smaller disaccharides, including sucrose, maltose, and lactose. This process is carried out by pancreatic amylase and brush border enzymes. The final step involves converting disaccharides into monosaccharides, galactose, glucose, and fructose, which are then absorbed into the bloodstream. A common issue related to carbohydrate digestion is lactose intolerance, which occurs when the enzyme lactase is absent or insufficient. This prevents the digestion of lactose, leading to gastrointestinal discomfort. By coordinating enzyme activity, bile salt function, and specialized absorption mechanisms, the small intestine ensures that fats and carbohydrates are efficiently broken down and absorbed, providing essential nutrients for the body. Carbohydrates are an essential source of energy, but before they can be absorbed, they must be broken down into monosaccharides, their simplest form. Digestion begins with amylase, an enzyme that breaks down starch and glycogen into smaller carbohydrate units. These are further processed into disaccharides, including maltose, sucrose, and lactose. Each disaccharide is broken down by a specific brush border enzyme. Maltase splits maltose into two glucose molecules, sucrase breaks sucrose into glucose and fructose, and lactase digests lactose into glucose and galactose. Once digestion is complete, monosaccharides must be absorbed into intestinal cells before entering the bloodstream. Glucose and galactose use a sodium glucose cotransporter, known as SGLT, which relies on sodium to actively transport these sugars into the intestinal epithelial cells. From there, they exit into the bloodstream through GLUT2 transporters. Fructose absorption occurs through a different mechanism. It enters intestinal cells through GLUT5 transporters and then exits into circulation via GLUT2 transporters. This process ensures that all simple sugars are efficiently absorbed and delivered to the body for energy production. Through the combined action of enzymes and specialized transporters, the small intestine effectively processes carbohydrates, converting complex molecules into readily available energy sources for the body. Proteins play a vital role in the body's structure and function, but before they can be absorbed, they must be broken down into smaller peptides and amino acids. Protein digestion is carried out by two main groups of enzymes: endopeptidases and exopeptidases. Endopeptidases, also known as proteases, cleave peptide bonds within protein molecules, breaking large proteins into smaller peptide fragments. These enzymes are initially secreted as inactive zymogens to prevent self-digestion. Pepsin, secreted by the stomach, is activated in acidic conditions, while trypsin and chymotrypsin, produced by the pancreas, function in the small intestine. Exopeptidases complete protein digestion by removing individual amino acids from peptide chains. These include aminopeptidases, which cleave amino acids from the amino terminal end of peptides, and carboxypeptidases, which act on the carboxyl terminal end. Once proteins are broken down, peptide absorption occurs primarily in the form of free amino acids, which are transported across the intestinal lining into the bloodstream. However, some larger peptides, such as dipeptides and tripeptides, can also be absorbed intact through specialized transporters. By utilizing proteases, exopeptidases, and specialized transport mechanisms, the digestive system efficiently breaks down dietary proteins into bioavailable components, ensuring that the body receives the amino acids it needs for growth, repair, and metabolic function. Proteins are composed of chains of amino acids, which must be broken down into smaller peptides or free amino acids before they can be absorbed by the small intestine. Protein digestion begins with endopeptidases, enzymes that cleave peptide bonds within protein molecules. Pepsin, trypsin, and chymotrypsin are key endopeptidases that break long protein chains into smaller peptide fragments. Next, exopeptidases act on these fragments. Aminopeptidases remove amino acids from the amino terminal end, while carboxypeptidases break down peptides from the carboxyl terminal end. These actions generate smaller peptides and free amino acids, making them ready for absorption. Peptide absorption occurs primarily in the form of free amino acids, which enter intestinal cells through sodium-dependent transporters. Dipeptides and tripeptides, however, can also be absorbed intact using specialized peptide transporters. Once inside the cell, these small peptides may be further broken down by intracellular peptidases before entering the bloodstream. Some small peptide fragments may be absorbed via transcytosis, a process where they are unclosed in vesicles and transported across the cell to reach circulation. This mechanism plays a role in the absorption of certain bioactive peptides and the transfer of maternal antibodies in newborns. Through the coordinated action of endopeptidase, exopeptidases, and specialized transporters, the digestive system ensures that proteins are efficiently broken down and absorbed, providing essential amino acids for tissue repair, enzyme production, and metabolic function. The large intestine plays a crucial role in water absorption and the formation of solid waste before elimination. After digestion and nutrient absorption in the small intestine, approximately 1.5 liters of liquid remains in the digestive tract out of the original 9 liters of total fluid intake. Most of this remaining fluid is reabsorbed in the colon, leaving only about 0.1 liters of water to be excreted in feces. This process ensures that the body maintains proper hydration while forming solid waste. The ileocecal valve marks the boundary between the small and large intestine, regulating the movement of contents into the cecum, a pouch-like structure that includes the appendix. The large intestine is divided into distinct regions: the ascending colon, transverse colon, descending colon, and sigmoid colon. Each section contributes to water absorption, bacterial fermentation, and stool formation. Two important structural features of the colon are tinea coli and haustra. The tinea coli are longitudinal bands of smooth muscle that create the segmented appearance of the colon. The haustra are pouch-like structures that allow for gradual movement and compaction of waste material. By efficiently reabsorbing water and compacting indigestible material, the large intestine plays a key role in maintaining fluid balance and preparing waste for elimination. The large intestine is responsible for the final stages of digestion, water absorption, and the elimination of waste. Motility in the large intestine is regulated by a combination of segmental contractions and mass movements. Segmental contractions continue to mix the contents of the colon, ensuring proper absorption of remaining nutrients and water. In contrast, mass movement is a powerful peristaltic contraction that moves the contents forward and triggers the urge to defecate. This is influenced by two important reflexes. The gastrocolic reflex occurs when the presence of food in the stomach signals the colon to increase motility, moving contents toward the rectum. The defecation reflex is triggered when the rectum fills with stool, leading to the relaxation of the internal anal sphincter, signaling the need for elimination. Although most digestion occurs in the small intestine, bacteria in the colon play a significant role in breaking down undigested materials. These bacteria produce beneficial substances such as lipophilic compounds, which can be absorbed, and short-chain fatty acids, which serve as an energy source for colonocytes, the cells lining the colon. They also contribute to the production of essential vitamins, such as vitamin K, and generate flatus, or intestinal gas, as a byproduct of fermentation. Proper water absorption is essential for maintaining hydration, and diarrhea can result in severe dehydration if too much water is lost in the stool. By coordinating motility, bacterial fermentation, and water absorption, the large intestine ensures that the final steps of digestion and waste elimination are efficient and balanced. The large intestine is the final section of the digestive tract, responsible for absorbing water, processing waste, and preparing it for elimination. Structurally, it is divided into several key regions, each playing a specific role in digestion and waste concentration. Food enters the large intestine through the ileocecal valve, passing into the cecum, a pouch-like structure that connects to the appendix, a small organ thought to play a role in immune function. From here, contents move sequentially through the ascending colon, transverse colon, descending colon, and sigmoid colon, where water is absorbed, and bacteria continue breaking down any remaining nutrients. Two unique features of the large intestine contribute to its function: the tinea coli and haustra. The tinea coli are longitudinal bands of smooth muscle that create the characteristic segmented appearance of the colon. The haustra are pouch-like structures that allow for gradual movement of waste material. The rectum serves as the storage site for feces. When the rectum fills, stretch receptors in the rectal wall initiate the defecation reflex, signaling the internal anal sphincter to relax. The external anal sphincter, under voluntary control, allows for conscious regulation of bowel movements. On a microscopic level, the inner lining of the large intestine contains intestinal glands, which secrete mucus to facilitate the passage of waste while protecting the intestinal walls. Additionally, lymphoid nodules within the intestinal walls play a role in immune defense, helping to prevent infection from gut bacteria. Through the combined actions of fluid absorption, bacterial processing, and regulated motility, the large intestine ensures that waste is compacted, water balance is maintained, and elimination occurs efficiently. The digestive system is a complex network that breaks down food, absorbs nutrients, and eliminates waste. We explored the anatomy of the digestive system, identifying key structures from the mouth to the large intestine. Each organ plays a specialized role in digestion, ensuring efficient nutrient absorption. Digestive functions and processes include mechanical digestion, chemical breakdown, secretion, absorption, and motility, all working together to convert food into energy. The regulation of gastrointestinal function involves the nervous and endocrine systems, coordinating enzyme secretion and motility. The cephalic phase begins before food enters the mouth, triggering salivary and gastric secretions. The gastric phase continues digestion in the stomach, where acid and enzymes break down proteins. The intestinal phase finalizes digestion, with the small intestine absorbing nutrients and the large intestine concentrating waste. Together, these phases ensure that nutrients are efficiently processed and waste is eliminated. The digestive system plays a vital role in maintaining homeostasis and overall health.